Okay, hello everybody. Thank you so much for joining me today and for attending this symposium. My name is Scott Ryall. I'm a laboratory genetics fellow at Brigham and Women's Hospital in Boston. And I'm going to be presenting to you today some of the work that I was fortunate enough to be involved in with my mentor and attending at the time, Dr. Adrian DeBuc, who has since moved on to the Roswell Park Cancer Center. I just want to emphasize that this work is, exclusively been done by him, and I'm just, picking it up at the end here and taking it across the finish line and happy to share it with you. So you'll see both of our contact informations on this slide as well as the last one if you are interested in reaching out to us and collaborating on any of the projects that I show today. So the title of my talk is Illuminating a New Path Forward for Cancer Diagnostics. So I don't think it can be overstated that cancer is a complex disease. And over the past 2 decades or so, since NGS became more commonplace, I think generally the genetics field is focused heavily on SNVs and small insertions and indels and things of that nature to really try to explain how cancer forms and how the tumorigenic process begins. However, that kind of ignores one side of the coin, specifically as it pertains to structural variants and copy number changes. So we can have broad losses and gains, which lead to polyploidy or aneuploidy, rearrangement such as insertions and translocations, as well as focal amplifications. It can be the result of double minutes or homogeneously staining regions. And for a long time, we've really kind of not advanced the technology we've been looking at in order to evaluate these things. Primarily, we focused on looking at these using a technique called karyotyping, which has been around longer than both myself and I'm sure many of you on this call have been on the planet. And the reason it's been used for so long is for one very simple reason, and that is that it works. But that being said, it is not without its limitations. Specifically, it requires dividing cells. There's a high rate of false positivity, and the most biggest setback for karyotyping is as a relatively low resolution when we're considering the complexities of the events that we may be detecting. Now, oftentimes, because of these limitations, the use of a karyotype often results in the need for cascade testing, some form of a confirmatory test that it confirms what you're seeing with your eyes. And in Boston, as well as in many of the labs that I'm sure you're in, this often takes place as FISH or chromosomal microarray. But again, these are also not without their limitations. Specifically for fish, this is a very targeted analysis. For chromosomal microarray, this is a genome-wide analysis, but it can't pick up things like balanced translocations. And when we look at some of the more modern approaches, and next-generation sequencing as well as RNA-based sequencing, there's a extra level of analytical complexity that is kind of added to the mix and kind of extends our turnaround times and makes things a little bit more challenging for us. So with that in mind, there's been a lot of emphasis and a lot of work looking into what is the better mousetrap. If you're a fan of Dragon's Den or sorry, Shark Tank, I'm Canadian, so Dragon's Den is the Canadian version. Laurie Grenier always says, can someone find a better mousetrap? And something that's very similar from the karyotyping side of things is, what is the combination of techniques we can do to identify structural variants in a reliable way? And I'm happy to report or happy to, kind of put up optical genome mapping as a potential solution for that better mousetrap. I don't want to belabor the methodologies going into OGM. I'm sure there'll be many talks at this symposium that go into great detail, but I do want to just emphasize a few points that are extremely important from the clinical perspective. The first is that this is about a 3 to 4 day turnaround time. And I always like to say this is a 3 to 4 day turnaround time until a finalized result. A karyotype might be faster. You rush a karyotype, in theory, it can be done in 24 hours. But that doesn't take into account the fact that there may be some cascade testing that has to take place, extra fish, extra chromosomal microarray, which all adds to the turnaround time. With optical genome mapping, in 3 to 4 days, you're getting a definitive result, and more often than not, a definitive result that's reported clinically. The other thing I want to just kind of emphasize is that, as I mentioned earlier with karyotype, there are relative issues in terms of the resolution limitations. However, for optical genome mapping, that's not the case. We get unprecedented levels of resolution down to about 500 base pairs. So, really, really powerful technique. And, when we first saw this, we were really interested in kind of applying this into our clinical setting and asking, what can OGM do for us and our patients? So when we started this project off, we wanted to look at kind of disease-defining structural variants that had matching karyotypes, fish, and RNA fusion data. So we had kind of these gold standards to compare it from, and we looked at kind of the hallmarks, things like BCR-ABL, PML, RAW, and things of that nature that are kind of disease-defining events, the tier 1As, if you will. And I don't want to spend too much time going through this, but what I can kind of conclude and what I want to emphasize is that we had 100% sensitivity and specificity. So anything that was picked up by our classical karyotyping or our fish was also picked up by OGM, which was, confirmatory to us and also really great to see it confirmed to us that OGM can be used to identify those things that are most important from a patient and our treatment perspective. And that included things such as the inversion 16. I don't want to spend too much time going through kind of each and every one of these, but I always like to highlight inversion 16. When I first started my fellowship, I missed an inversion 16, so it kind of haunts my dreams a little bit now, and is always one that I find extremely challenging to see. But you can see here in this case, you see the karyotype over here in the corner. Optical genome mapping has no problem identifying it and highlighting the CBFB MYH11 translocation. And the same can be said for things like the KMT2A rearrangements, PML-RAWA, as well as BCR-ABL. Again, kind of those really important disease-defining alterations that optical genome mapping really doesn't struggle with identifying at all. But with that being said, I didn't want the emphasis of my talk today to be on look at all the fun things that OGM can find that we also find with karyotyping. I think where optical genome mapping really kind of can flex its strength is in those more complex cases, those instances that kind of, keep us up at night, make us pull our hair a little bit, and are a little bit more confusing. And I want to highlight a few of those cases with you today. This first one here is an NPM1 IDH1 mutated AML that was in a 56-year-old male. And when it came to us, we identified this inversion 3 event, which you can see in the red box, as well as this short deletion on 11q, sorry. But we were a little bit curious because the patient wasn't responding as we would expect in the clinic. So we ran optical genome mapping on it. And what that revealed to us was that although our karyotype was correct in what we were seeing, the level of complexity that was actually happening under the hood of this case, we severely underestimated. You can see on the side here, we had about 25 different structural variants including a four-way translocation, which involved 3 and 11, and a lot of other events that we simply couldn't see because they were cytogenetically cryptic. And that's going to be extremely important moving forward because, as we all know, genetic complexity is an influencing factor in both prognosis and diagnosis of different myeloid malignancies. So we're really going to have to take, into appreciation what we mean by complex, especially when we're using these techniques that are able to kind of refine all of these, nuanced changes that we may not be able to see with our eyes on a standard karyotype. Here's another interesting case. This is a 39-year-old male that came to us and presented in the clinic with kind of classical CML symptoms. The bone marrow biopsy and the aspirate both kind of clear-cut CML. Everything was saying CML. It got sent to our molecular team. They did the qPCR for the P210 and the P190 transcripts, which you likely do at your lab as well. But both of those came back negative. So the clinician reached out to us in the cytogenetics lab and said, listen, it looks like a CML. Everything fits with CML, but I'm not getting BCR-ABL transcript positivity. What's going on here? So we ran our karyotype, and you can kind of see down here. This is a complex karyotype, but the two things I want to draw your attention to are the two red squares, where you can clearly see the 922 translocation, that kind of classic puffy pale region on the terminal 9q, and the short Philadelphia chromosome from the derivative 22. However, with that being said, we wanted to kind of confirm this. Obviously, when our molecular disagrees with our karyotype, we want to be sure, and we ran our fish on it. And we did see the fish pattern that we would expect to see. You can see these fused signals occurring, which are indicative of the derivative chromosomes. But one thing we also noticed consistently, and one thing that our fish tech reached out to us about was said that, for some reason, one of the fused pairs was always a little bit too far apart. And you can see representation of those I'm highlighting with my mouse here. And although they're within the detection limits of our assay and we're able to be assigned as positive, it just didn't really sit right. So we wanted to take this one step further and run optical genome mapping on it. And as you can see, as a kind of a consistent theme, the specimen was significantly more complex than we may have originally appreciated. But what I will draw your attention to immediately is that we do have a 9 to 22 translocation, but it's actually a three-way translocation that is also involving chromosome number 2. And what's even more impressive about what optical genome mapping was able to do is it was able to tell us what the transcript itself was. And this was an extremely rare transcript, a P203. I'd personally never seen one before, extremely limited evidence of this in the literature. But essentially what this P203 transcript involves is exon 13 and exon 3 of ABL. So the issue being with our molecular testing is that where both of these are detecting P210 and P190, which as we know make up about 99% of our transcripts, the reverse primer in ABL was sitting in exon 2, not in exon 3. So our test wasn't able to pick it up, but OGM was able to quite nicely. And where this becomes extremely important is that our clinician was extremely hesitant to start a TKI on this patient without confirmation of the BCR ABL. With this information, they were confident in starting them on the TKI, and the patient's doing wonderfully and responding great, and is behaving in the way we would expect a CML to behave, which was a great story. The last case I'll show you in this part of the talk is again a kind of a classical CML that came to us again, kind of presenting like a classical CML. When the karyotype came to us, this is what our result looked like. Again, that kind of classic 922 translocation, that nice puffy terminal 9q, and that short Philadelphia chromosome. Our fish confirmed it was a 922 translocation, so everything kind of got the check marks and was ready to go. But that being said, once again, our molecular did not detect it. So that was a bit bizarre to us, but again, with the karyotype and the fish, we were confident in saying that this was a 922 translocation, so we were able to start the patient on a TKI. And about 2 weeks later, the clinician reached out to us and said, "Listen, you guys need to double check your karyotype in your fish because this patient's not responding to the TKI at all. The white blood cell counts are still sky high. They're deteriorating. Something's up. Something's not working out great." So we then ran it on optical genome mapping, and what we were able to see was that there was indeed a 922 translocation, but it was about 9 megabases away from BCR enable. So it wasn't a BCR able translocation. It was a 922 translocation that didn't involve either BCR or able. But what actually this patient had was a KMT2A ITD, which was what was driving their disease. Which then ends up begging the question, how much do you truly trust your eyes? Here are three sets of chromosomes that kind of have that classic 922 translocation appearance. However, only one of these is actually a 922 translocation. Everybody always asks, so I'll tell you it's this one right here. And this introduces this idea of a chromosomal mimicry. So, chromosome morphology that mimics a known structurally relevant variant, but actually isn't that variant. And, I'd like to say that these are extremely rare events, and they are, thankfully, for all the clinicians who sign cases out, it'd be terrifying if 50% of these things that we're signing out may not be real. But at about 1%, these aren't insignificant. They are going to constitute a small percentage of the cases that we're dealing with, and understanding that and using classical karyotyping without something like an OGM to refine it is going to be extremely challenging and is going to limit our clinical utility of the results that we sign out. So to summarize our myeloid experience so far, optical genome mapping was sensitively and specifically able to detect the disease-defining copy number variants and structural variants, which is again a really good basis on which to build, or a foundation, sorry, to build, future testing. Is that whether or not you're using this only for refined, difficult cases, or just standard cases coming, every case that comes through your door, it's going to be able to handle the variants that are driving these malignancies. Optical genome mapping was able to uncover structural variants that exist, and I'm air quoting this, below the surface. So these are things that we don't see karyotypically, but kind of really speak to the complexity of what's going on, in these malignancies. It also helped reveal unexpected limitations of our current diagnostic approach here in Boston. So understanding that our molecular test was, detecting most of the events that we could have seen in that P203 case, but not that specific variant. And the use of OGM was what actually allowed this patient to go on a TKI and to kind of reap the benefits of that modern treatment approach. And leading into this is that the optical genome mapping does have real clinical consequences, and it can be extremely valuable in your cytogenetic workup. So I want to shift gears a little bit now and finish up this talk by just talking a little bit of some of the new data that we're working on right now, specifically working in multiple myeloma. And anybody who's listening to this who's worked in multiple myeloma knows that, in essence, working with these is extremely challenging and it's not fun. And the reason for this is that it often necessitates some form of a plasma cell enrichment before being able to analyze. And most labs are either doing this by some form of a flow approach, or in our lab, we're doing a CD138 enrichment. And if any of you have done this before, you know that the CD138 enrichment is not high throughput. It's technically challenging. It's time and labor intensive. It's just overall not a fun process to do. And, oftentimes I remember sitting with Dr. Dubuque and just saying it'd be so convenient if we just didn't have to enrich. And that kind of spurred this question about what happens if we can't. Can OGM kind of eliminate the need for an enrichment in these plasma cell neoplasms? So we took a small cohort of 10 multiple myelomas that ranged in plasma cell counts between 2 and 16%. We ran them under the standard workflow of the optical genome mapping, and it came out with nothing. We essentially got absolutely no indication of any variants going on, likely just representing the fact that we just didn't have enough plasma cells in order to detect these. But that being said, you don't need to run this under the standard approach. In theory, the maximum theoretical coverage of these flow cells is about 500 gigabases. Um, And what that translates to is about 1,000x coverage, not 300 to 400x coverage. So we essentially tweaked our approach and we asked ourselves, can we actually get to 1,000 coverage? And this took a lot of in-lab kind of tweaking and altering our protocols, really kind of precise control of our input, and really taking care when we're doing the extraction process to make sure that we maintain that really long length of DNA fragments. But what we were able to see is that when we started enriching in about, 8 to 10 nanograms per microliter for the labeling section, we were able to consistently get up over the 1000x coverage. And we've been able to replicate this moving forward by using this kind of refined approach that I'm happy to share with anybody who's interested. And what we noticed was that once we started going to 1000x coverage and really letting the machine run, all of a sudden these events started popping up. And this, for example, is the 1114 CCND1 IGH variant, which was seen at about 11% variance allele frequency. Again, this is without enrichment. So again, a disease-defining event that we're able to identify in a specimen that was not enriched. This is likewise another situation where, again, another case that was not enriched where we're able to identify a TP53 deletion, which is likely contributing to the disease. And it also works for things like hyperdiploidy. So you can see here this kind of classical copy number approach with trisomies in 3, 5, 7, et cetera, et cetera. But what's also extremely interesting with this patient is that it also was able to identify another CCND1 IGH translocation that was kind of happening in the background of the hyperdiploidy that, realistically probably would have been missed, if it wasn't run on optical genome mapping, as most of us will do some form of FISH approach to identify these hyperdiploidy cases. And this is the last multiple myeloma I'll show you, and it's just again another instance where the clinician came to us and said, "Hey, this is a multiple myeloma. It has the 1114 translocation. We started them with venetoclax. They're not responding great. What's happening?" We were able to look a little bit closer into these three locuses, which are sitting over top of BCL2. And if I zoom directly into locus number 3, you can see this is right over top of BCL. You can see this immense complex event happening here, but essentially ends up resulting in a copy number gain of about 10 to 12 copies of BCL2, which has now been recognized as a mechanism of resistance to venetoclax. And we're not exactly sure why or what's specifically causing this amplification, but again, it is revealing to us some different mechanism that we may not have, originally considered for this case, and kind of provides an explanation for what we're seeing in the clinic. So to summarize our myeloma experience, optical genome mapping, at least in our hands so far, has been able to eliminate the need for plasma cell enrichment, which is fantastic from a lab management standpoint and a turnaround time standpoint. It has the potential to detect clinically relevant structural variants and expand the understanding of the structural variants. So things like that BCL2 amplification, kind of really understanding what causes these elements of resistance or these different disease mechanisms. It can help identify background events that are a little bit less salient. That's a good example with the hyperdiploidy case that had that 1114 in the background that likely would have been missed if it wasn't for optical genome mapping. And as I mentioned, it can help identify these non-canonical mechanisms such as the BCL2 amplification. Now, as I did mention, this is kind of work in progress. We are still working on putting this together as a publication. I know several groups will be interested in the ability to eliminate the plasma cell enrichment for multiple myeloma analysis. We are still evaluating the limits of detection. You might have noticed in the case samples that I showed, they often came in with a little bit higher plasma cell counts between the 10 to 20% range. We really want to know, can we do this technique when they come into us with, the 2 to 3 to 5% range, which is what I personally find we often get when they're submitted to us. So we really want to be working on that. So we're working on dilution series to see really how low can we get the plasma cell counts in order to get them to be able to be detectable, with the optical genome mapping. We're going to continue to evaluate the sensitivity and specificity over a larger cohort. Again, we only have about 10 or 15 multiple myelomas so far, so we obviously want to expand that, which leads into my next part. We're looking to evaluate between 50 and 100 to really better understand the structural landscape of multiple myelomas that I think for a long time has been a little bit neglected just because of the challenges with analyzing them. And most of us just kind of default to some sort of a fish panel to kind of, figure out the pertinent changes that might be causing the malignancy. So with that, I just want to thank everybody that was involved in these projects that are listed here. Sam Brody's the tech that runs all of the optical genome mapping. He's extremely talented, so we're very lucky to have him. As I mentioned, this is work that I conducted with Dr. DeBook, who's now at Roswell Park. Please feel free to reach out to either one of us if you have any questions or concerns or are interested in collaborating in any way. And I just wanted to end by thanking Bionano. It's really been a pleasure working with them. They're one of the companies that is, very actively involved in the process, really receptive to any troubleshooting that you may come into. And it's been really a pleasure to work with them in the optical genome mapping and to hopefully get this clinically implemented at our institution. And with that, I'll thank you all for listening and enjoy the rest of the symposium. Thank you.